Ok, muy buenas tardes ya a esta altura, así que muchas gracias por venir. Para mí en realidad es un placer estar aquí y presentar a Sebastián, el doctor Sebastián Glim, me dijo que había que pronunciar la I alargada, así que Sebastián Glim, él es especialista de Science en Alemania, en microscopía, y vamos a tener una charla que se marca en un curso que nosotros hemos realizado esta semana, de hecho está en curso, termina mañana, de bioimágenes. Ya, en realidad ha sido todo un éxito entender, aprender y ver cómo todos nuestros estudiantes, tanto nacionales como extranjeros, que han llegado a nuestra facultad, han descubierto las grandes potencialidades que tiene las bioimágenes, cómo nosotros podemos capturar datos, cómo podemos trabajar básicamente con estas imágenes y entender el trasfondo y el fundamento de las imágenes. Entonces, esta es una charla complementaria al curso y doy las gracias a Nicola Araya, a Felipe Pérez de Arce, por su apoyo de Microx, que ustedes vieron afuera, tienen un pequeño stand, ellos nos han apoyado en este curso, así que les agradezco su colaboración. Ya, sí. Bueno, sin más, les dejo entonces la palabra a Sebastián Glin. No sé, por favor, Sebastián.
Muchas gracias. Yeah, thank you so much for these kind words of introduction. And I hope that you can all actually hear me quite well also in the back. If not, okay, so that's great. So welcome to that discussion that we are going to have today about microscopy evolution. And the question that I'm starting with is actually whether AI and automation can really help us to lift microscopy and also image analysis or post-processing to the next level. Because we all know that AI is basically growing exponentially, and all the tools that we can use nowadays for our daily life or for decision makings are actually driven by automated processes and also by AI. So what we could maybe imagine, and maybe we go one step further, so not just answering whether AI and automation can help us, but maybe just imagine the future in a couple of years where we have some robotic lab assistant that is basically telling us, or telling your boss, so basically the head of the department, that you should focus your research on a specific topic because AI has evaluated publications and actually finds out, okay, maybe you need about 2 years to accomplish a project or 3 years. So in 2 to 3 years, what will have a strong impact? What will give you a very good publication? What are maybe the proteins that you should investigate to get a good publication? And of course, a very efficient publication, because every one of us wants to have a good work-life balance. Yeah? So we should not do unnecessary things that are maybe not of interest in 2 to 3 years. So AI can really help us to filter and prioritize. But what is important for us is actually the question, and also for the professors, is the question, can we really trust these machines? Can we trust artificial intelligence to make these decisions and to help us with making these decisions? And to answer that question, let me do a little detour, so a little bit away from sciences or from natural sciences, and answer that question from a more philosophical point of view. So what is actually the time point when we really trust a new technology in general? And one can actually answer that with 4 main points, which is, on the one hand, the technology needs to appear useful. Yeah, that's of course something that is intrinsic, but this is really necessary. And we need to feel comfortable with the new technology. If there's something out and we're not really feeling comfortable to use it, then we're not accepting it. And of course, it needs to be easier than before. And of course, the learning curve. How much time does it take to learn and adapt to a new technology? Yeah? So what needs to be in the end, it needs to be a positive outcome. So you need to save time with the new technology. So basically, the effort learning the new tool should be less than the effort that you save by this new technology. And this is actually not something that is new. This acceptance or this technology acceptance model that I now outlined in a couple of words, this was actually published already in 1989, the year where the Berlin Wall fell. So some of you probably have not seen that in person. So I actually was living just 500 meters away from the border. So for me, this is still a very vivid experience that I made. And Davis actually described that in this very nice house where a new technology is basically the foundation, but whether it will actually get a roof depends on the two strong pillars of perceived usefulness of a new technology for your daily life, for your work, and also the perceived ease of use. And to illustrate that these two columns are actually very important for technology adoption or technology acceptance is if we now translate this into something that everybody of us uses and probably has experienced, which is the acceptance that a phone, a mobile phone, is actually not anymore just a phone. Because what a phone actually originally was a device to call somebody, to speak to some person and be reachable wherever you are. But nowadays, or actually not nowadays, actually, just since the year 2007, that's what I predict now personally, when the first iPhone generation was released, the people, they perceived this ease of use. Before, there was usefulness with smartphones from Nokia, from Ericsson, many companies that are not existing anymore, that are not producing smartphones anymore, because they had good technology, but the ease of use was not there. The ease of use was just introduced with the iPhones in 2007, and then all the others jumped on it or basically died, like BlackBerry, etc. So these two different columns are important, the usefulness and the ease of use. And nowadays, as we all know, we use our phones, our smartphones, not only to make phone calls, but to play games, to find new partners, and do some shopping, order food, but also work, answer emails, or use some applications to do many things that hopefully fulfill our life. But what the system is also doing nowadays, and what we are often not really aware of, is that the smartphones, the applications, they actually help us, or maybe not help us, but they go into the direction where they lead us to make certain decisions. Yeah, so they intrinsically and either unconsciously or consciously, they help us with decisions. And whether they're helping or not is maybe something debatable in some philosophical class. They also influence our political attitudes. Yeah, they are not only influencing what lifestyle we want to have, what clothes we want to wear, but if you're listening to certain videos of certain influencers and they have half a sentence or so speaking something bad against the current government or some other politicians, you are actually driven into some political directions. And I think every one of us has to answer that question, how far we want to go, how critical we have to think, so that we are still staying the master and not completely be driven by the smartphones and the applications and the algorithms, basically. Okay, so it comes down to the question, okay, do we really trust that technology? Do we want to trust it, or do we simply accept it, that this type of new technology gives us good directions for our career, for our work, for our life? And that's something that really depends on all of us, what applications we use, to what extent we use it, and how critical we are. But we accept it, yeah, because it's easy and it's useful. Now let's make the switch back to science, yeah. And in science, my question is, why not accepting a robotic lab consultant that can help us? Because we are struggling nowadays with so many different questions, different issues like publication pressure. We are not often having enough money to fulfill our research. We have time limits because we want to have a good life apart from our academic life. We might have some kids. We want to have a good work-life balance with our families. We still want to stay connected with other colleagues. And some of us also have to do some teaching. So we have some teaching obligations. So, and all of that we need to put under this big umbrella, which is called life. And life gets more and more complex, and the good thing is that life also invented some technologies like AI to help us a little bit to get out of that, this kind of vicious cycle, so to say. So the big problem for researchers, at least from the perspective how I see it, is this increased complexity. Nowadays, it's not anymore possible to just publish a finding based on one technology. So you cannot just take a few pretty pictures with a microscope and publish that in Nature. That is not possible anymore. That was possible maybe 30, 40 years ago. But nowadays, you need to prove your findings with different methods. So you have to be kind of a little expert in all the different technologies that are available to actually prove maybe pictures that come from a microscope and that are validated, for instance, with a biochemist approach. And also where we take a look at the single technologies within that complex multi-methodological environment, we also have an explosion of possibilities. 30 years ago, we maybe had just 10 or 5 different types of microscopes, different general types of microscopes from which you could choose from. And nowadays, there's an explosion of possibilities even within one technological area, which also makes it more complex to get your head around what technology do I use, how do I use it, the best way to really prove my findings, to then not work more than I actually have to, so to work efficiently, to have a better work-life balance, and so on. And this leads to the situation that you have lots of knowledge to process, and of course, the pressure is not decreasing, it's rather increasing in terms of times, publication, etc. So it's becoming more and more difficult, yeah? And that leads to something that we actually call, or not we, but what we also see in the literature, is this so-called reproducibility crisis. So nowadays, there's so much data being produced by so many people all around the world, and reviewers often don't really have the time to critically look at some data. And when you or colleagues, etc., try to reproduce some findings, quite often it's not really possible. So the data that you actually get out is maybe appearing not so much trustworthy. And I think most of us, and I actually hope that 100% of us, want that their data is trustworthy, so that what we get out is really the truth. Yeah, but if you cannot reproduce it, it might not be the truth. Yeah, so you need to have a good understanding about the technologies, or use the technologies in a sensible way, so that the data is reproducible. So when we now again go back to microscopy, and translate all of what I just told you to basically the pain points that microscopy users have, but also microscopy supervisors, lab technicians actually have, is that for a microscopy user, I told you it's a very complex environment. So what you normally do in life when things get complex, what do unfortunately also many people do when political things get very complex, they tend to go for easy answers. They tend to go, and that's the only last sentence that I say about politics, they tend to go for the easy answers given by certain political streams, yeah, because otherwise it would be too complicated to get your head around certain scenarios. So what you would do in the microscopy field is you would try to just use the buttons or the microscopes that you are comfortable with, and the rest you would really leave behind, because you don't have time to understand how it works. So what is the big problem? Not too much for the microscopy producers, but for research in general, is that you could do so much better if you would simply know how to do it. And normally for this, we have the microscopy supervisors. But the microscopy supervisors, they typically face the challenge that they have lots of different microscopes that they have to supervise, and they probably have lots of students that they need to train. And they have students that they have, or some results they have to evaluate, they have to do processing, image analysis. So there is not enough time actually to provide this so-called best user experience for all the microscope users. So that's a big issue. Yeah, so you could do so much better and you want to do so much better, but we have developed into a direction where the world got so complex and where we simply need some help to be more efficient. So it was already about 10 years ago when some people published a couple of nice articles in scientific journals about microscopy and the need to have smart microscopes, so systems that help the user to achieve better results. Because already in 2015 or several years before, it was already said by many professors all around the world, look, all my PhD students, master students, postdocs, they don't have the time to become microscopy experts anymore. The microscopes, they need to become easier. Yeah? Otherwise, I don't get my results, I don't get my publications in Nature, I don't get some funding for the next generation of the microscope that I want to buy from you. So you have to help me to get better results so that I can buy the next generation of a microscope. And what we all have to accept, also the older generation, is that nowadays we don't want to have a steep learning curve anymore. When we install a new application on our smartphones, what we want is, we want to understand it within 2 minutes. If we don't understand it, we typically delete it and we go for the next application and hope that this is more intuitive to be used. So that is something that all the societies worldwide actually face, that something needs to be more intuitive. And if you sum up some smart microscopy features that are wished by the scientific community, it's of course the ease of use, but automated options to set up the microscope, integrated microscope support, reduction of training time so that the facility managers or the lab technicians have more time to support more people, help with image analysis, and something that is actually coming really like a super fast spaceship, other topics, machine learning and artificial intelligence. That was not on the plan 10 years ago when these publications came out, but about 2 to 3 years ago, people really see that machine learning and AI is something we have to really watch out for. So if we summarize that now into different chapters, and that's what I'm going to do now, we can maybe start with some features and some technologies that are already available in the field of machine learning and AI. Because what we actually see is that when we take a look at all publications that are being released and correlate that to publications where to a certain extent machine learning or artificial intelligence technologies were used, we actually see that these two curves are strongly converging. So there is more than half of the publications and even more publications where we actually have already machine learning and or artificial intelligence included in the publications. So let's just wait until they took their seat. So what you find with many microscope producing companies, and I'm not just showing you what we from ZEISS actually have, is when you go to the different websites and you're just Googling or searching on the website artificial intelligence, machine learning based topics, at least from ZEISS we have a very active community. We have many postdocs that came to us, to our foundation, and are working on that topic AI, because we realize that you are saving very much time by the usage of AI and by the usage of machine learning. And if you want to give it a check, at the end of the presentation I have some QR codes which you can scan and then be directly directed to the website, and then you actually see internal discussions, what our product managers, developers think about it and what approaches we actually take to tackle the topic artificial intelligence. And what I can already promise is that this discussion is a very lively discussion, because we at ZEISS, we are in a very luxurious situation that we are not a company, we are a foundation, and we spend 15% of our revenue, not of our profit, 15% of the total revenue, we always spend into research. So we are having many, many ideas which we are typically following up on. Several do not really make it. but we really try to be very open-minded, and that's one of the big plus points, I would say, with the ZEISS group. Okay, but let's talk a little bit about AI and structure it, because many people have learned or have used the term AI and machine learning and don't really know what's the difference. So AI is basically the big umbrella that is around the big topics of machine learning and deep learning, which are parts of the big umbrella artificial intelligence. So basically anything artificial, yeah, that is generated from software output can be called artificial intelligence generated. And if you basically do some decision making based on some predefined algorithms, this is then machine learning. So you have predefined algorithms, you ask the software to make a decision, but based on specific algorithms. And then the machine basically learns to make a decision of something. Is this maybe a hand of a human or is it a hand of a monkey? And then the system learns based on certain parameters. And if you allow the machine to make individual or kind of random crosslinks between the different algorithms, so basically you enable it to think by itself. So maybe I have 2 more loops in this direction, 3 like this, but completely randomly, then we call that deep learning. Yeah. Okay, and this is something, one of our first results that we actually have and which we actually give out as a product to help users is our AI-based sample finder or AI-based sample detection tool that we have. So we have a tool which helps the users to put the sample into the microscope and also to scan the sample and then eventually identify your structures, your region of interest that you want to scan later on, and also automatically defines the area for the final experiment. So you don't have to search for the sample anymore, and you don't have to set up some regions that should be scanned. So this is already done automatically, and we call that AI sample finder. So it's machine learning basically, and we trained, I think, in total 25,000 slides that were coming from different users to really identify where is the cover slip, for instance, or where is the petri dish, etc. Differentiate the sample from, let's put it like this, suboptimal sample preparation artifacts like air bubbles, dirt, fingerprints, etc. So this all needed to be trained on, and yeah, this was done by many, many students at the University of Jena. Okay. What we also have, and this is actually just being released in 2 weeks, we also have a co-pilot inside our Zen software. At the moment, it is now released for the confocal systems that we have, but it's basically a ChatGPT functionality as a part of our microscope software. Because we realize that when you have a question nowadays, or at least for me since about a year, I'm regularly using ChatGPT. When I have a question, I, for instance, want to know what's the difference between Tesla Model Y and Volkswagen ID.7. What are the USPs for this and this cars? What are the difficult things? I'm not Googling that anymore. I just ask ChatGPT for that question. And that's what you can also now do with a microscope system. So you can basically say, as it is printed over here, how can I improve the quality of my image? And the system, or the ChatGPT functionality, is taking a look at your hardware. What hardware do you have? And then is proposing you some solutions, what you can do to get a better image quality. And you can really get to ChatGPT or to our microscope copilot as with ChatGPT and really continuously ask some questions to really go down to where you actually want to go. Then we have something else. We also have machine learning for image analysis. And we call this product from us Intellisys. That's just our name for it. And it works quite in an easy way if you want to segment different groups of objects, like background, mitotic cells, or just cells that are normally in the cell culture. It's a very easy tool. So usefulness and ease of use, that is something that we need to have to adopt and accept a new technology. And here it is just with a small brush, just painting a few pixels, and then the system is training itself to understand what are mitotic cells, what are just the normal cells, what is the background maybe. And then you can plot that in a nice time plot. So it's very easy to be used. Or what we have now, and here we actually do something else. We are doing something that is called instant segmentation, where we have some AI in a project where you need to separate objects that are close to each other. That's always a difficult part. And this AI tool is actually finding the cells automatically. And if they are not doing a super good job, you can just use the brush tool and just add a little bit more and increase its intelligence. Yeah? But this is something that is done in the cloud because this requires lots of computing power. So the training of the model is done in the cloud, but you can download the model and then apply it for all of your data. Yeah, but the training needs to be done in the cloud. And this is just an example for some geological applications where you have different structures that show a different color scheme and polarization. Yeah, and as you see, it needs to be useful, it needs to be easy. Yeah, so these are the two aspects that the new technology has to have. But there's always a but. Yeah, so with everything in life, unfortunately, there's a but, and the thing here is it takes time. Yeah, and that is also what we see everywhere. So when you want to have fast AI processes to be executed, you need to have very strong GPUs on board, or you need to buy some let's say GPU power online in different cloud formats, and this is really the bottleneck nowadays. Yeah, so either you have enough time and then it will be done within half an hour, or you say, okay, it is also okay to wait for 2 days or so for the model to be trained, and then it's not so costly. So time is money is the big but over here. But I think that is with every new technology. The good thing is that you don't have to buy it, you can just use it online on demand, and then it's done, and you don't have to invest, I don't know, 20,000 euros for a super expensive computer, which you only need maybe for 1.5 year. You just log on, you upload some data, you train your model, you download it, and when you don't need it, you also don't have to pay some money for it. I will show you this QR code also at the end of the presentation, so that's a little guide on how to do artificial intelligence for image analysis in a relatively efficient way, but also tells you a little bit about the pitfalls, so give you a little bit an input on critical thinking about artificial intelligence. Next topic, open development and collaboration. What we actually see quite often is that people all around the world try to collaborate, or they find something in a publication which they would like to use from another user, or people would like to share it across the continent or even across the university. And what we as a foundation have actually managed is we have built up a cloud-based solution. We call it Arrivis Cloud, or the predecessor version was called Appear, where you can basically upload your scripts for your image analysis or try out scripts that are provided by us for free or by some technological partners, and then share these scripts. So basically, I have a very nice script in MATLAB that I'm uploading into the cloud, and I can basically say or give access to specific persons to use it, or even grant the download of this. And the only thing that the person that downloads it has to obey is that the author needs to be mentioned in the publication if you get some positive outcome. And the good thing is I can generate my MATLAB script in a very old MATLAB 5 point some version, and you basically download it on your system and you will run it in a Docker container. So you don't need to have the MATLAB license. Yeah, it will be run in the Docker container, and all the licenses, this is paid by the Zeiss Foundation in the cloud. Yeah, that is a really cool solution. And what we also do via this solution is we basically provide solutions to any type of partner. Could be an industry partner that wants to have a specific software solution for image acquisition or image analysis. And we basically shuttle back and forth data and scripts until there is the perfect solution. And also when you have a specific problem and you cannot really solve it by yourself and you need some programming assistant, we at Zeiss also provide that. Not for free, of course, but you can also send the request and then upload some data, and our people in our software center in Munich, they will evaluate how long will it take, how much will it cost, and then you get some specific solution. Another big topic, as we already discussed it a little bit, is the field of automation and smartness. So one part of that is automated image analysis, and you have seen some movies already how this can be AI-based or at least AI-assisted. And when we talk about automated image analysis, there are these two big fields, I would call them, which are individualized image analysis, which could be AI-based, and really automated image analysis, which could also be AI-based or just threshold-based, for instance. The automated image analysis has the big plus that it's super easy to be used. It's maybe just one mouse click. So in our software, we call that the bio apps. So it's basically a couple of modules that we provide for very easy answers. How many cells do I have? How many genes are expressed? How many spots of something do I actually count in my nucleus? So it's just one click and you get the results. But it is not so specific. So that's the downside of it, yeah? Because it is a model that is trained to work on multiple different sample preparations. So it works to a certain degree, which is the case with all microscope-producing companies that offer these single-click solutions. But they work, of course, best if you fulfill a certain workflow. Yeah? If you use specific chemicals, then certain microscope companies, they guarantee you, then our one-click solution will always work. We from ZEISS, we go a different direction. Although we have some solutions like this, we are not exclusively focusing on that because our idea has always been, and it's basically imprinted in our DNA, that we don't want to restrict the researchers. We always want to have flexible solutions, which is speaking a little bit against this ease of use approach. Yeah? But what we want is we don't want to put some boundaries in your head. When you do some research, you should freely think, and we try to offer you solutions that are for this purpose as easy as possible. Okay, what we can also do, of course, is we can basically scale up image analysis. So what we have, but you probably also know that you could have a server somewhere at universities, you could have some free-floating licenses with image analysis, and everyone who just needs the image analysis license modules, they can simply just have it and execute it on their own laptops, on workstations, computer, etc., so that you can really parallelize image analysis. And also when it comes to more high throughput, high data image analysis, we have different software solutions, and these are in the field of Arriva's image analysis solutions located, where you have this software located on one workstation computer, which acts as a server, which can be accessed by multiple people, and you can have some pipelines, and maybe person one starts some image analysis. On Monday, 2 hours later, another person puts another request into it so that you have a constant execution of image analysis processing workflows going on. Next topic, automated experiment setup features. And this is, for instance, something that we call guided acquisition workflows. It goes into the direction of what I showed you already, that the machine is automatically detecting your tissue. But what you can also do already is you can use machine learning, you can do different type of individualized image analysis and basically scan your sample and then really with machine learning teach the machine to find your samples automatically. Yeah? So even if it is maybe different from the ZEISS initial AI sample finder solution, you can then really individualize it. You can even say, scan my slide or scan my plate and only find my cells or structures of my brain slices that show colocalization of specifically 2 colors. And then the system finds these regions of interest and just extracts the coordinates and then, for instance, is doing an automatic super resolution Z-stack. So the idea is that you need to invest some time to train the machine to do that, to set up your experiments and your image analysis. But in the end, you do your sample preparation, you put it on the microscope, you click one button and you go and have a better work-life balance. Yeah, that's basically the idea of it. Yeah, so the machine will then really go to all the coordinates automatically, focus, do the picture image acquisition, do some processing if this is necessary, and then you come back and you have the result. And the big plus here is that you are saving lots of time, But the other big plus is that you're saving lots of data, because you don't have unnecessary data that you need to evaluate or shuttle from one place to another place. So again, you save time, you're more efficient, you have maybe more time for your life or for other obligations. And then what I showed you already is help functionality like ChatGPT that we have actually built in. So it was just a few years back, we started with many videos that we uploaded in our user portal where you can really see how to do certain things. And this is now actually already becoming a dinosaur technology because of ChatGPT functionality that we actually have built in, where you can really just ask the Copilot some questions and get some responses back. But this at the moment still relies on internet connection. Yeah, so it's not put onto the computer because we are regularly training it so that it becomes more intelligent, so to say. And also we tried that it really understands different kind of slangs, because we know that not everybody, not even me, so we are not fluent in English, so English is not our mother tongue. So if a person from Chile, Germany, etc. Writes something in with maybe wrong grammar, our Copilot should nonetheless understand what is meant. Yeah, and I think we got to a relatively robust solution already. And this, as I said, will be rolled out in I think 2 or 3 weeks, so beginning of May, this will come out. Yeah, so and this, I guess, will help all the facility managers to reduce the training time, yeah, because the users can simply do that by themselves. Yeah. Okay, so next part is how can we help with setting up the hardware automatically so that everyone, no matter how much knowledge you have, actually gets an optimal image. Because it's not just the software that you need to set, sometimes you also need to set the hardware. Lenses, position the lenses in a good way to have a sharp picture, to not have, let's say, some color fringes when you go quite deep into the tissue, or that you have maybe some automatic barcode reading in the beginning, or has some calibration of your sample or your glass slide or your multi-well plate that is put in. As I said, adjust the lenses for plastic, thick plastic, thin plastic, glass. There can be so many different scenarios, and there needs to be some automated tool to do that. And AI will not do that. AI will basically help with software solutions and evaluate maybe images, but the hardware settings, how to optimize the hardware depending on your sample, requires some different degree of automation or intelligence. And that is basically something that we have built into one of our first smart microscope systems, that is the cell discoverer system. It's a live cell automated microscope system that can really adapt itself to the different samples that you have, so that you will always get a good picture. And what we have also built into the system, it's unbreakable. So whoever works with the system cannot break it, which is also one of the biggest fears of a facility manager, that some people break it or leave the system unclean. You have to clean it, or when it's broken, you have to wait for maybe a couple of weeks until it gets repaired, and you have to find some money sometimes. Sometimes the lenses cost $20,000 US dollars, and then the system is not usable. Nobody can use it. People have planned to do some experiments, prepared some animals for that, and that is something that should belong to the past with such a microscope system, which is protecting itself and helping all the users to get the same image quality. In such systems, we can also integrate, of course, into some complete robotic streets, where you have robots taking some plates, putting them into the system, taking it out, putting it back into the incubator, putting it into some staining device to wash in or wash out certain compounds, to do multiplex imaging with maybe up to 50 different colors. So this is all possible nowadays, and we're not producing the robots by ourselves. We have a collaboration with a British company, PAA, and they actually provide robotic solutions. So this is just a microscope, but here's a robot, and there's an incubator, there's some washing street, there's a hotel where one can put some plates. And this is just a real-life example where such a system is actually put into some of this completely automated imaging and staining street. And why do I show you this? Not because I want to make advertisement for you to buy such a system, but because you have such a system here in Valdivia already. Yeah, so you got that 5 years ago, just in the beginning of the pandemic, where you have this lifestyle imaging machine, which can be used for lifestyle applications. But what you have also purchased is this fancy robot. If you have some fixed endpoint assays, so fixed samples in plates or dishes, you can put them all on this little hotel rack. I forgot how many you can actually put on, but quite a few. And then you can, with the scheduler software, say what plate to scan when, and just have a repetitive scanning of it. Yeah. Okay, the other big topic that is also important to consider in the field of microscopy evolution is connected microscopy. And what do I mean with connected microscopy? I told you already that nowadays it's not only enough to take some pictures and publish it, but also to prove your findings with different technologies. But what is also important is to put it in some context. Yeah? So what people want and what you like to read when you read a publication is to see the big picture. Where does your finding fit in? Yeah? So really go from macroscopic observations actually down to molecular findings. That would be the perfect publication. So if you really have a publication that contains everything, shows the context, shows the relevance, start with the disease, etc., and then go down and use multiple different technologies down to the molecular level, that is where you get the Nature publication, or at least a high impact publication. So what you see, what we offer, except for the smartphones actually, we offer many different lens-based systems to really go from organism or organ-based observations down to molecular and protein observations. But what we now then want is, we want to connect all of the data. And for that we have a program, a software solution, it's called Zen Connect, where you can then put all of your data together in one project, align it, and then generate movies or images that show you how the different worlds, the macroscopic world, the microscopic world, the nanoscopic world, are connected. Okay, and of course connected microscopy could mean that you have everything connected in your facilities. So all the different systems that are connected to a centralized data storage server, then the microscope workstations where multiple different people can work on different systems remotely or live, directly on site of the system. You have offline workstations to do the image analysis, either live or also remote accessible. And what we have, what is really strongly used, especially in the Asian countries, is that you view your data and that you view your data processing results with mobile devices. So you're basically at home or in the tram, etc., and you take a look at it and you maybe share the pictures. You send it to your collaborator, you give permission to your boss and say, hey, look, this is what's the outcome right now. Yeah, what do you think is good? And then they could maybe use their smartphone, the tablet, to do some annotation and say, oh, maybe look at this region when you rescan or when you re-image your sample. And the last topic, maybe not too much important for the majority of you, but for the microscope supervisors, which is automated maintenance. And to make the relevance of that topic appealing to you, some of us actually, we have a car. And what we don't want is that the car breaks down and we don't have a car, because we often need the car to get somewhere. With a car, what we typically have is we have maybe an inspection every year, or every 2 years. But what about if we would have some artificial intelligence that is, or some kind of service that is running in the background that is basically checking whether there might be a problem coming up. Yeah? And that is something that you could easily find out with many technologies, because for every technology you have specifications that a technology has to fulfill. When I click on a button in my smartphone, the application has to open within maybe half a millisecond to 0.6 milliseconds or seconds. And if this is not okay, then we need to do something about it. Yeah? For the user, maybe that's not a big problem, but if this continues and nobody is doing something, at one point the application might not work. And that is something that we have actually built into our systems. On the one hand, that, for instance, hardware components can automatically check themselves to to produce always the same outcome, like for instance our LED light source, which is always checking itself how much light is coming out, because we know that any lamp will degrade over time. And what you want in research, you want to get the same pictures with the same experiment 1 year after you've acquired the first picture. So you don't want to have any differences. So that is something that we provide. And what we also provide is something that we call predictive service, where the machine, the microscope, or not all, but many microscopes can already check themselves for specific actions, like rotating the filter cubes, rotating the objective turret, etc., and is measuring how long this takes. And this information is then actually then stored on the acquisition PC, and either, if you allow it, automatically sent to some Zeiss service web page. And when we see, or when the program sees that certain actions are delayed, or do not really work how we expected, then there's automatically a message sent to our service colleagues. They will double check on the report, and then they will contact the local people and say, look, we actually found out that there is maybe something coming up with your system. It might be good to schedule some service appointment. And which is also quite good for some facilities, some sites, where you don't have a service agreement and where you basically need to pay for that. Because if something breaks and you first need to find some money, this prolongs the time where nobody can use the microscope system. So this predictive service helps you to have the better user experience and to have the system really running 24/7. Okay. So, what's the conclusion? Let me come to the end of my discussion. So, I guess all of you might agree or will agree to my sentence that automation and AI definitely helps us to reach our goals faster. And what we have to simply accept is that we should never really give basically the decisions out of our hand completely. So we should always remember who the master is behind everything. But there are so many things that are already available with microscope systems nowadays, and there will be even more, because I said the topic AI and automation is exponentially increasing also within the microscopy producing companies, we are not actually lifting our experience to the next level in the future, but we have already done that. We are already on a very steep inclining track to achieve that goal. And let's do a little time warp to the future in the end. So when we all sit here together, maybe by the year 2050, or maybe even earlier, I could imagine it's maybe 2030, 35 already, and we're asking us the question, do we remember the times when microscopes just magnified objects? We probably will all laugh about that, like with the smartphones nowadays, because these were just the old dinosaur times when it was just about the picture, but not about the other issues. So, thank you so much for your attention, and here are the QR codes that I promised to you, and I'm happy to get some questions from you. Thank you so much.
Thank you very much, Sebastian, for a really nice talk. ¿Tenemos tiempo para algunas preguntas? Sí. Perseguir. Isabel.
Thank you very much for your speech. In your experience, do you believe that AI and microscopy intelligence will become cheaper in the future and reduce boundaries in the countries that are developing, or in smaller university or something like that? Or do you believe in contrary, it's going to be expensive to those countries or those universities get access them? Mm-hmm. And something like that? And do you believe that we are close to getting cheaper, or are we closer to getting more expensive to get access to all this intelligence and all of this AI and microscopy intelligence? Thank you very much.
So it's a very good question. And if you would have asked me the question maybe 3 weeks ago, or actually 2 weeks ago, I would have told you I believe that it will become cheaper because the production of the GPU cores and all the facilities where the processing is actually going on is heavily increased and the prices dramatically drop. But with the tariffs that Trump actually imposed and the new trade wars that we have worldwide, I think this will be a serious topic to be considered because most of the production is in the Asian countries or has been moved to the Asian countries. And if there's no release of these products to the Western countries or release of certain components to the Western countries, we then get to a similar situation that we had a couple of years ago with where we had this shortage of cars because there were not enough semiconductors being produced. So now there are strategies from the United States, from Europe to build up their own semiconductor production lines so that you're not so much dependent on certain countries. But if you're doing that in your countries and if you're not following capitalism basically to go where it is cheapest, then of course the prices will increase again. And what we also have to consider, and this is I think very important topic for us in Germany, is all the calculations, all the online-based model trainings, AI-based processing. This requires lots of energy, lots of electricity. And it depends on the strategy of your country, whether you make that available for a rather low price or what directions you go. So I don't know whether you know, but in Germany, for instance, we shut down all of our atomic power plants, we shut down all of our coal power plants, because we really want to go 100% renewable energies. And I think we are one of the only countries, if not the only country in the world that is doing that. But nowadays we face this issue, how do we manage with all the energy that is consumed by the internet to do all the calculations with all the electrical cars that we want to have? Because I think you also know in 2035, no combustion engine is allowed to be sold in the European Union anymore. But there are clever solutions. For instance, I don't want to make advertisement, but there are new solutions, for instance, coming from Tesla with energy storing devices. BYD is producing very good LFP batteries to store energy, because a big problem that at least what we see from the calculations in Germany is not that we do not have enough energy or renewable energy, but we don't have the energy at the right times. We have lots of wind energy during the night, but we're not using the energy during the night. So we need to save it somehow, and we cannot pump up some water onto the mountains because we don't have so many mountains in Germany. So we need other clever solutions to do smart, maybe AI-driven energy management in a complete country. And if this is then working out, it could become cheaper. Or if you subsidize, for instance, atomic energy, like France, for instance, they have, I think, more than 50 atomic reactors, and they heavily rely on nuclear energy, then you can also drop the prices. But I think it's a difficult question. It's a complex answer to your question.
Gracias. Alfonso Smith, nuestro profesor del curso.
Thank you very much. Just talking about or thinking about the reproducibility associated with AI and automation, because basically it will be associated to your technology or your criteria, how you like size, thinking about the scientific community, because basically reproducibility will be associated to your technology and not to the complementary technology that is in the market.
That's true. So when it comes to the reproducibility, so first of all, coming to the models that are used, for instance, in our AI technology, most of the models that we use, we of course adjust them to our systems, but that are coming from the Google Foundation or from the Google company. So when it comes to image analysis, etc. And basically, we are giving you the tools, and you are basically generating your own trainings for your data. So we're not giving you, let's say, we give you also these one-click solutions to get solutions out quite quick. But with respect to AI and image analysis, you are still the master in terms of training your model and verifying the result, whether the result is really what you believe in. That is your question.
But what's happening with someone that wants to reproduce what I'm doing?
So then they probably have to ask you, please give me your model and take a look at it. And therefore, we actually have that cloud service where you can exchange the models. Exactly, this is accessible. So the cloud solution was an idea from our foundation where everybody could actually access it for free. And now I think you have to pay some money depending on how much gigabyte you actually need to use. But that's really the idea, to exchange the data, exchange the scripts to really prove that your data plots are actually true or not.
Super, thank you. ¿Alguna otra pregunta? Si no, thank you very much, Sebastián.
You're welcome. Thanks for the invitation again.
Eso. Muchas gracias por asistir. Chao, chao.